Now, when we think about an antibody, we normally think of this Y-shaped structure, which to a large part is true. But it's actually found in only 3 of the different types of antibody classes. And let's quickly mention these just so it's clear in our heads. It's normally found in IgD, it's found in IgE, and it's found in IgG. And our other antibody classes, IgA and IgM, they actually have slightly different structures, which we'll talk about in another video. So the structure can be split up in different ways. And the first way that we want to look at it is through the chains of the antibody. So let's label these first of all. So we've got our heavy chain, which is this middle part of the Y shape, this longer part that goes through the middle. And then we've got the light chains on the end, on the external part of the antibody. So that's the heavy chains and the light chains, 2 of each within each antibody of these classes here, IgD, IgE, and IgG. Okay, so a heavy chain can be further split up into 5 different parts, whereas a light chain can be split up into 2 other categories. Let's go through the heavy chain first of all. We've got the mu heavy chain, which is normally found in IgM. Now we've got the alpha heavy chain, which is found in IgA. Delta heavy chain is found in IgD. We've got gamma heavy chain found in IgG. We've got epsilon heavy chain found in IgE, right? So that's the heavy chains. And what I've just mentioned there is the class of antibody. That's the 5 different classes. 5 different classes of antibodies, which remember can be shown or depicted by this acronym MAG, right? That's an easy way to remember the 5 classes of antibodies. Now you might be wondering, what is the difference between a heavy chain and a class of antibody? And that will be a very valid question. A class contains one and only one type of heavy chain. However, for example, it can have either type of light chain. So if you've got IgM, it's always going to have the mu heavy chain, but it can have a lambda light chain or it can have a kappa light chain. Okay, so hopefully that's clear in our minds what the difference between a heavy chain and a class is. And we mentioned the types of light chains. There's 2 types. There's lambda and then there's kappa. Okay, cool. So now that's clear in our minds, we can move on to the next part, which is our variable region and our constant region. And these are found in both the light chain and the heavy chain. Variable regions tends to be obviously found at the top, depicted by the light blue region, and the constant region is found at the bottom, the second half generally of the light chain, and the bottom 3/4, if you like, of the heavy chain. Now, the variable region is the area in which the amino acids can change depending on the antibody that's coming from a specific clone of plasma cell. So a specific clone of plasma cell is going to produce an antibody which is specific for only one antigen. This bit at the top is called the antigen binding region. That's the antigen binding region, which is pretty self-explanatory if you ask me. That's where the antigen binds on to the antibody. Okay, now let's take a look at the light chain and heavy chain and the different parts and actually label these. So the light chain, first of all, at the top in the variable region, you have your actual variable region, which we'll label as VL because it's part of the light chain. But you also have this joining region, which is also part of the variable region, found right at the bottom. So we'll label that JL. And then we've got our constant region, in which the amino acids are all the same, and that's CL. So that's our light chain. The heavy chain is slightly different. At the top, you also have your variable region, VH this time. Then you've got something called your diversity region, in which the amino acids are also very variable, DH. Then you have your joining region, JH. In terms of the constant regions of the light chain, you've actually got 3 constant regions. So you've got CH1, you've got CH2, and then you have CH3. So there's 3 constant regions within the heavy chain. Okay, now the last part here. We've got another classification, if you like, of the structure. based on the top part of the antibody and the bottom half. The top half and the bottom half, okay? Now the top half is what we call the fragment, that's a fragment antigen binding region. That's the fragment antigen binding region, also known as the Fab region for short, okay? And self-explanatory here, guys. The antigen binds on to this region, and that's the purpose of the top half of the antibody, if you like. And when we look at the bottom half of the antibody, this part is called the fraction crystallizable region. That's the fraction crystallizable region, which is known as the Fc region for short. And this part of the antibody, the lower half, is responsible for the effector functions. So what that means is that the lower half of the antibody binds to different proteins and starts different processes within the immune system and links different parts of the immune system together. For example, it can bind to complement and activate complement. It can bind to a macrophage and activate a macrophage, and similarly bind to various other proteins. But there's one little part that we left out earlier on, and that's this part here, which is the hinge region. And as you can see, this is the part of the antibody which forms a Y shape. It's where the antibody actually bends. And as part of the hinge region, you'll notice small S's in the middle. And we'll just draw an arrow and label these. These are disulphide bonds. That's disulphide bonds. And these bonds are a combination of covalent and non-covalent bonds which link different parts of the antibody together. So you'll notice in the hinge region in particular, there's 2 disulphide bonds, whereas other parts of the antibody, for example, from different parts of the light chain and different parts of the heavy chain, or different parts of the heavy and light chains linking together, there's going to be a single disulphide bond in place. So there you go, that's a quick overview of the structure of an antibody.